think I'm back. All right. So the other 22 pairs are what we call autosomes. Okay. So 22 pairs that are not determinative of biological sex are called autosomes. So there's, again, pair numbers 1 through 22. And then there are pairs 23. Those are the sex chromosomes. So just, again, we're biologists, we name everything. We're biologists, we name everything. All right. Now, next up, want to be sure that we've got things like, yeah, want to be sure we've got terms like haploid and diploid down. I did show you this earlier, but I want to be sure that we've got this down and also the symbols for it. So when you see the symbol that it shows 2N, that means this thing called diploid, and you can see the dye. And all that means is you've got both sets, okay? You've got the cells in your body, the regular old cells, basically not the sex cells, not gametes, okay? That show both sets of chromosomes that are there. You've got one set from mom, one set from dad, and again, that's every cell in your body, your somatic cells, okay? Those have diploid cells. And so look, here's one copy from your mom, one copy from dad. So here's mom's, here's dad's. And this is chromosome, I'm making it up, number 1. Here's chromosome number 2, and here's mom's, and here's dad's, and you received that. This is in your body, I'm making it up. And then chromosome 3, and again, mom, dad. These are homologs, okay? Now, haploid, half. Think about it like that. It is represented by an N, and haploid, N, half. Here again is chromosome number 1. There's only one of them though. Chromosome number 2 and chromosome number 3. The only place you would find those like that is going to be an egg or sperm. That is called a gamete. The only place you find those are in gonads, ovaries or testes. Okay, that's where they are produced. And that is via meiosis. Okay, and that's what we're gonna eventually be talking about today. All right, now each set of 23 chromosomes contains 22 autosomes and a single set, usually typical, single set of sex chromosome. Now, in an unfertilized egg, the ovum, the sex chromosome is X. Okay, so again, this is typical. And in a sperm cell, the single chromosome may either be X or Y. So I want you to think about this. Okay, let me do some drawing. Okay, so if you are a mom, again, biological female, you've got 2 X's in your ovaries. You're going to undergo meiosis. So this one cell right here, okay, is going to do a double split into meiosis. Basically, your sex cells are going to land, they're going to replicate, and then they're going to split twice. All you've got to offer for your eggs that you're going to give your offspring, you're going to make eggs. The only thing you have to offer are X's. So you're going to make eggs that have X's, haploid ones, right? These are just going to be N, just haploid. Okay, that's all you got. Here you go. I got some eggs with X chromosomes in them. That's it. That's all you got. Okay, now think about this. If you're a biological male and you have gonads with XY, because you're biologically male, okay, when you go to make sperm, you've got X and Y. So when this replicates and then double splits, I'll show you that in a second, some of your sperm cells are going to have X's in them because you're going to replicate that DNA. Don't forget that. And some of your sperm cells, well, that looks like an X over there. Some of them are going to have X's. This is Y. That's a Y too. And some of them are going to have Y. Now, these are going to randomly join up with eggs, okay, which already have X's in them. So this could be XX, this could be XX, this could be XY, this kid could be XY. So do you see where I'm going with this? So who determines the sex of the offspring? Gals, all you have to offer is X. Guys, you're the only ones with a Y chromosome. So you are the ones determining the sex of all the offspring. 50-50 shot. You've got some sperm with X's in them and you've got some sperm with Y's in them. Remember that Henry the Eighth guy? He was killing all of his wives. Remember that guy? Because they were producing only girls. He was quite obviously not a geneticist because it was his fault. Now, kind of already gone over this, but let's just put a diagram to it, shall we? So gonads, and remember, dudes, you do not have the corner of the market on gonads. Ovaries are gonads. Testes are gonads. It's just any organ that produces sex cells. And so gonads produce haploid, half the genes, half the chromosomes, let's say that. Haploid gametes, okay? So these gametes are formed via meiosis. So if it was just mitosis, it would just be clones. And if this cell had 46, mitosis makes it'd be 46. So that wouldn't work because then 46 in an egg, 46 in an egg, you would end up at 92. That would not work. So you want your eggs or your sperm to have 23, 23. So an egg meets sperm, you come together, and the kid has 46, just like you. And so you maintain the chromosomal number, which I'll show you here in a second. So you want this meiosis, remember, fertilization, back to meiosis, back to fertilization kind of cycle. And so gonads produce that. So meiosis is occurring in the gonads, and that is where this double mitosis is going to occur that is going to lead to this haploid production of gametes. We're going to maintain our chromosomal number. Now, look at this diagram. This diagram is showing us, I'm going to switch to green. Okay, so this is showing us a little bit of follow kind of the red and blue here. Red for mother and then blue for father. So like mitosis, meiosis, and again, notice the spelling, is preceded by replication. So anytime that we are going to undergo cell division, you've got to replicate first, okay? And when we replicate, remember, you go from a single chromosome and then we're going to replicate it. And we've got now the centromere, the little belt holding together your sisters. So anytime you see this thing right here and this little belt right here, there's your centromere holding together your sisters. Okay, that's replicated DNA. It's not supposed to look like that. Not on an average daily. It only looks like that after it went through S phase, okay? After it went through the synthesis. So it's like, hey, you need to get ready to undergo cell division. Same stuff, okay? All right. So here's the key. 2N in this case is 6. This is a very strange organism, okay? So there's only 6 chromosomes in this organism. Very weird. There are 3 maternal and 3 paternal. So 1, 2, 3. 3 paternal, 1, 2, 3. Here's homologs. Here's homologs. Here's homologs. Okay. Now, in this particular one, you can see that everybody is replicated and they're held together by their centromeres. So we're getting ready to undergo meiosis. Heck, this could be meiosis as well. You don't really know. All right, so you've got this replicated and you've got two identical sisters. Okay, but same for meiosis. Now, meiosis is a double, like I've been saying, is a double mitosis. The idea here is that it's actually a reduction of the number of chromosomes. You go from diploid, which is every cell in your body, even the ones that start out in your gonads, and you go from diploid chromosomes to haploid chromosomes, okay? And we're gonna call it something very simple, thank goodness. We're gonna call it meiosis 1 is the first set of divisions, and we're gonna call it meiosis 2 for the second set. Nice and easy, okay? The nice thing is they're both following PPMAT, okay? And at the end, you end up with daughter cells, just like the first one. But in this case, okay, because you go through it twice, instead of ending up with 2 daughter cells, you're going to end up with 4 daughter cells, okay? And because again, you go through it twice, and please note, you do not undergo replication between the two meiosises. There's probably a different word for that. Between meiosis 1 and meiosis 2, you cut that number, that chromosomal number in half. Okay, so if you look at this is spermatogenesis, that means creation of sperm. Okay, so if you look at that down here in the diagram, you start off with 2n46, okay, and then you go to 23, you split once, and then you split again, and you end up with the haploid number, 23 in each one of them. And you're like, that looks a little weird because how can you go 23 and then 23? Well, there's a sister issue. So I'll show you how that works here in a second. Okay, so let's look at this really quickly. So I want you to write this down. I want you to put it on a sticky note. I want you to, I don't know, make a Quizlet card. I don't know what you got to do. But this is what I want you to try to remember about the two types of meiosis, okay? So in meiosis 1, you've got a separation of homologous chromosomes. In meiosis 2, you have a separation of sister chromatids, okay? You kind of need to memorize that. And then after you have that memorized, you can walk yourself through PPMAT twice. You just have to know who separates in each phase. After you know that, no problem, because it's mitosis 2 times. We're going to throw in something for fun here in a second. It's actually kind of cool. But you just need to know who separates first and who separates second. It will become very obvious here, like what has to happen. But when you're learning it, you really need to learn that meiosis 1, homologs separate, meiosis 2, sisters separate. In that order, okay? So it's reductional first and then equational. I don't really care if you know that, because I think you just learn homologs first, sisters second, okay? And I'll show you what it looks like. And then we can, again, talk about the why and all of that good stuff. Okay, let me show you right here in this diagram. So in meiosis 2, 2 stages. You're going to start off with interphase, just like you always do, okay? And that's where you're going to have replication. It's going to go through the S phase, okay? Now, prophase 1 is the big difference. And I'm going to go over a whole section on what prophase 1 is. But notice you've got prophase 1, like you basically got, and then this is PP mat, but we're shortening it. But you've got, and we're just calling this 1 for each of these, and then you've got 2 for each of these. They're pretty much the same. Okay, you just got to remember homologs and sisters, all right? At the end of this, you've got 2 cells. At the end of this, each of the 2 cells split, so you get 4 cells, okay? But again, it really is the same. Here, the only difference is the homologs. Everybody lines up on the metaphase plate, and then the homologs separate. And then again, we line up on the metaphase plate, and now the sisters break their centromeres, and they pull apart. That's the big difference. So memorize that. If you do that, it will save your life for meiosis. So homologs, then sisters. Homolog, sisters. Homolog, sisters. Okay, this is what it looks like. So here's your single chromosomes. Remember, that's normal. The sister thing is not. That means it's replicated, right? That's not real normal. Okay, so this is interphase. And then everybody's going to replicate. So your homologous pair replicates, and now you've got sisters held together by the centromeres. So that's just interphase. You've not done anything yet. So, and you've got, only 1 pair of chromosomes here. Then meiosis 1 occurs. At the end of meiosis 1, that's 1 homologous pair, 1 pair of chromosomes right there. So at the end of meiosis 1, PP mat occurred, and you have the separation of homologs. Look, they're just, the homologs went to, cytokinesis, went into opposite cells. You're like, okay, but that still looks real weird, right? Okay, so no replication, no interphase, nothing else happens. We're going to go right into meiosis 2. And remember what happens in meiosis 2? Sisters are going to separate. So we're going to line the sisters up on the metaphase plate now, and we're going to break those apart, cytokinesis, and then I think that made that thing happen. And then you've got 4 cells with single chromosomes, okay? And note what happened, okay? You've now got separation. So up here in this first cell, you had mom and dad together, and now you've got these replicated dudes, and everybody's in a single cell. These are haploid, right? There's not 2 chromosomes in there anymore. So those are now haploid cells, unreplicated. Everybody is all lonesome, but this is haploid. And so now those can become either egg or sperm, depending on what gonad that happened in. Okay, so this is meiosis 1. Okay, I'm going to talk to you about that. I'm going to leave this BioFlix for you to watch on your own. Okay, I always joke and say it's mildly pornographic. It's not, but that's my favorite joke for class. So I'm going to let you've got a link to it. You can watch it. I do recommend watching it. It's actually really good. It explains PP mat times 2. Okay. But in meiosis 1, it's preceded by interphase, and then there's replication into sisters, right? So that's the whole point there. And the sister chromatids are genetically identical, and they're joined by the little belt, holds them together. Okay. The single centromere replicates, and it forms 2 centrosomes. Okay. All right. So you've got your centrosomes getting ready for your mitotic spindle. Okay. Now, here's what it looks like. Okay. So remember how I told you we just name it and thank goodness, but it's like prophase 1, metaphase 1. So, and then prophase 2, we like this. So it looks really, really, really similar. And thank goodness. Okay, and it's super easy. So prophase 1, we're gonna spend a whole bunch of time on here in just a second, but everything else is crazy easy. So in prophase 1, we have the creation of the spindle, the nuclear envelope dissolves. We have, this crazy thing that happens called crossing over, and you have this chiasmata form. I'm going to show you in a second, but this is the hardest part of meiosis. But it's also the coolest part. It's why you and your brother and sister, or sisters or brothers, are not identical twins, even if you were born at different times. It's why you're all different if you have the same two parents. And then once that crossing over happens, then you just have metaphase. Those homologs line up on the metaphase plate. In anaphase, look, the homologs, they're still sisters, but the homologs separate and they go to their opposite poles, and then we split them equally. Okay, now look, we have two cells that come out of that. Now we're entering, going straight into meiosis 2. Okay, meiosis 2 starts with prophase 2, and prometaphase is here. They're just being simple about it, thank God. But it's the same thing. And you have, the dissolving of the nuclear envelope. Here comes the formation of your mitotic spindle. And look, here you have what's left. Your sisters are lining up on the metaphase plate. And now your sisters separate, and now each of those cells divide, and you end up with 4. And these are haploid daughters, okay? And there are 4 of them. That's kind of like the nuts and bolts, like real quick and dirty. All right, now meiosis 1, kind of let's zero in here. Meiosis 1 occurs in the same 4 phases, in the same 4 phases that you've seen before. Now, we want to focus in on prophase 1. So prophase 1 is the most important part because it's different. It's the only thing that's really different about meiosis. Prophase 1, in fact, even is the longest period for meiosis at all. It's like over 90% of the time that's required for meiosis. So the chromosomes begin to condense, and then they're going to do something kind of crazy. They're going to go through this process called synapsis. Now, here's the important thing. So you know how those homologs were kind of like all lined up, and then they line up on the metaphase plate, and then the homologs separate in meiosis 1? Well, before they separate, they're in prophase 1 and they're lined up, okay? While they're lined up, they will align very specifically. So the chromosome number 1 from your mom and the chromosome number 1 from your dad, they line up really specifically, loci to loci, gene to gene, really specifically. And then what they do is they form what is called a tetrad. And that tetrad is this process called synapsis. They line up and they form this tetrad. So all four of them are together. That's the tetrad. And you can kind of see it in this diagram right here. And then each tetrad, usually, it doesn't have to, it's random, can form these little X-shaped regions. So if there's all four of them and they line up, some of them can form these little X regions where they cross over like this. And those little X regions are called chiasmata. So here's 2 chromosomes. They are homologous, and they can cross over like this, forming this little X thing. And those are called chiasmata. Craziness happens now. These non-sisters, okay, will actually swap that chunk of DNA. They'll swap it. That is called crossing over. They'll exchange those DNA segments. So like your mom's chunk exchanges it with the dad's chunk. Crossing over. It's called crossing over. And what that does is we're increasing the genetic variability of the egg or of the sperm, okay? This is the foundation of genetic variation, and why you are different than your sister if you're female, why you're different than your brother if you are male, if you have the same parents. Like you're similar, right? Because you came from the same parents, but you're not identical twins because of this. It takes a while, okay? So synapsis is a process where the homologs line up, they form tetrads, there are 4 of them. They make little X's, chiasmata, and then they swap chunks. Anywhere from 1 to 4 of these usually, and it could be 0 really, because it's random. It doesn't even have to happen. It could, might not. And this takes a long time, 90% of the entire meiosis time. Now, and meiosis two, well, let me go back to meiosis one. And then meiosis one, the rest of it is really just lining those homologs up on the metaphase plate and then separate it. An anaphase, you're just separating homologs, and then cytokinesis, telophase and cytokinesis. Very, very, very straightforward. Nothing's really all that different. Okay. Then meiosis two. Meiosis two is very, very, very similar to mitosis because you're separating sisters. The only difference here is that the sisters have some weird genetic differences. See in the photo how you have like some of the blue chromosomes, like dads have weird little red tips on them, and vice versa. That's because crossing over has occurred. And so here, again, lining up sisters on metaphase plate, breaking the centromere and pulling them apart in anaphase. And at the end, you've got 4 cells. Okay, so at the end of meiosis 2, you've got 4 daughters, but big difference is they are haploid and they're unreplicated, right? They're just single chromosomes. And so they are a mixture of some from mom and some from dad. Because if you look at some of those diagrams, if you were kind of like paying attention to the metaphase plate, some of those chromosomes from dad and some of those chromosomes from mom were lining up on different sides of the metaphase plate. It wasn't like all of mom's lined up here and all of dad's lined up here. It's random. And so they got pulled apart and now they're in different places. And so there's this weird combination, okay? Random combination in each egg or sperm. And so they are very genetically distinct one from the other, which nearly guarantees you unique offspring every single time. Okay, so when you compare mitosis and meiosis, here's kind of the gist to remember. Mitosis conserves the number of chromosomal sets. So if you start out with 46, you're supposed to end up with 46. And they're clones, they're genetically identical. Meiosis, you are reducing the number of chromosomes. So you go from diploid, undergo 2 sets of division, and you go down to haploid. And the whole idea of this is that you differ genetically. So you're trying to get some differences in that particular cell. And really, each and every cell, you're trying to make different genetic combinations. Now, this is a nice diagram just to kind of help you look at the 2 types of cell division. So again, really look at mitosis versus meiosis. A lot of the differences, well, there are a lot of differences, but a lot of the differences prophase 1, look at, in meiosis crossing over is going to occur. And remember, meiosis is a double cell division. Obviously, mitosis, there's only 1 cell division, and in that one, just sisters separate. In meiosis, you've got homologs separating first, and then sisters separate after that very long phase in prophase 1 of the crossing over occurring. And so there is also some descriptors down there in the summary. So please look at that, and that might help you distinguish some things. I will ask you questions about please distinguish between mitosis and meiosis other than the spelling. All right, now genetic variation is obviously key to success of living organisms. It leads to things like evolution. It leads to things like Darwinism and natural selection, and again, therefore, evolution. And so, how do we get genetic variation? Well, I just kind of explained one of those ways, but let's kind of start to pinpoint some of these because later in the semester, we are going to be talking about Mendelian genetics. We're going to be talking about ecology and evolution in populations, and we need to kind of understand the foundation. So, one of the ways you get genetic variation in the populations through the sexual cycle is through mutations. Now, a lot of times we think mutations are like, you drop a, Peter Parker in the, vat of radioactive goo and he comes out as Spider-Man, right? It's not exactly how it works, although it makes for a good comic book and a lot of movies. Make a lot of movies. Mutations are really about changing individual A's, G's, T's, and C's in the genetic code, in the DNA. If you look at the diagram here, I want you to look at the original genetic sequence up at the top. And then I want you to look at the same genetic code, but with a mutation. So mutations can be caused by all sorts of things. Sometimes it is just exposure to something, kind of bad for you. You would call that you can call it like a mutagen. Sometimes if it causes cancer, it's called a carcinogen. If it causes birth defects, it's called a teratogen. I mean, but a lot of it still comes down to mutation. And so look at the second line. See where it should be a T, and then it has been mutated, and the base is changed to a C. Now, that is one type of um, DNA mutation. And I want you to think about what that causes, okay? In this particular case, what that would cause is a different amino acid to be inserted into the polypeptide chain, and therefore it would completely change the protein that is produced in this particular area. That can have pretty severe consequences, and sometimes it wouldn't have any consequences, but most of the time it does. Now, you can also have additions. See how that C is coming in? Now, what this is called is a frame shift. Also, the same thing would happen with a deletion mutation, where see how you're losing that thymine. Now, frame shifts occur where the reading frame, and we haven't talked about that quite yet, but I want you to imagine again how you read this and how it gets coded into amino acids that get built into polypeptides that get built into proteins. This will completely change the protein that is generated from this, completely changes how the RNA is made, and completely changes what the RNA produces. So these are all significant, but mutations can be usually are deleterious, which means they're bad. Okay, usually the organism does not survive, but sometimes when they are beneficial, it can cause what we consider rapid genetic change in a population, but that is fairly rare. Okay, it's fairly rare in general. Now, independent assortment of chromosomes. So this is a Mendelian thing, and we're going to talk about it a little bit later. But remember how I said, hey, did anybody notice how different moms and dads' chromosomes were lining up a little bit different on the metaphase plate, and how you didn't always have like dads just here and moms just up here? Like sometimes it was dad, mom, dad, mom, dad, 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 mom. Yeah, that kind of thing. This is independent assortment. Okay, it's random. And so what you end up with is a different combination of dads and all of his family lineage and mom and all of her family lineage in different gametes. So you know how somebody says like, oh, you look more like your dad. Oh, you look more like your mom. This, okay? You can technically end up with only your dad's chromosomes in his that side of the family in a sperm cell, okay? Or only the mom's side of her in her egg cells. So it is possible you can look more like your maternal lineage or more like your paternal lineage kind of idea. Possible. Okay, so this is independent assortment. Here's where it gets a little bit daunting though. So get your math on, everybody. So The number of combinations possible, I'll read it to you. The number of combinations possible when chromosomes assort independently. So this is like lining up the chromosomes for humans on the metaphase plate and then split. One goes this cell, one goes that cell. Into gametes is 2 to the power of n, where n is the haploid number of your chromosomes. So how many chromosomes do you have? What's your n number? Your n number is 23, like you have 23 pairs, right? That's your haploid number. So that means this is the number of possible chromosomal outcomes. So how many possible eggs or possible sperm could a human make just by lining up in different ways on that metaphase plate? It's 2 to the power of n. So if you take 2 to the power of 23, and you can load it into your little calculators, okay, and hit the button, it is over 8 million possible combinations for every egg and every sperm. That's a lot. That's why we all look so different. So here's kind of what we mean by independent assortment. So this is just, there are 2 chromosomes here, everybody. So 2 equally probable arrangements of chromosomes at metaphase 1. So again, metaphase 1 here, and we're obviously ignoring prophase 1 for a second. So look at, you've got all the paternal and maternal lining up on one side, and then here's just a mixture of the 2. And here's metaphase 2, where we had, here we split homologs. Okay, homolog went here, homolog went there. Homolog went here, homolog went there. And then here we split homologs, but look, they just lined up differently. So homolog, homolog, but look, you've got male, female, maternal, paternal, okay? You've got maternal, paternal. And now we're going to do it again. So in the combinations that are produced, look at what happens. You've got all paternal in this egg and sperm, all maternal, all maternal. Over here, though, they're all mixtures of mom and dad. Both of those are equally likely, equally likely. And that's independent assortment. And now imagine, and this is also interesting just to point out, like this organism is a fake organism, is 2. And how many chromosomes does it have? What's the haploid number? 2. So 2 to the power of 2. So 4. So it's got, okay. All right. So I'm going to show you a little bit about crossing over. This is an antiquated joke, but does anybody remember that show from way back in the way back? There was this guy, John Edwards. He had the show called Crossing Over. And he was like, he could talk to dead people. It was on the television. I don't remember when, but I would tell that joke and everybody thought it was hilarious, but now nobody knows what I'm talking about anymore. Okay, crossing over, when again, you've got the chromosomes and they do synapsis and then the chiasmata form and then they swap bits of DNA, that produces what we call recombinant chromosomes, okay? So the recombinant chromosome thing and crossing over starts really, really early in prophase 1 when those homologous chromosomes line up. And remember, non-sisters trade chunks. Wouldn't make any sense for sisters to trade chunks because they're just copies of themselves. So non-sisters, moms and dads, swap, okay? And what it does is it contributes in a huge way to genetic variation, okay? Into this idea that you are creating now bigger amounts of variation into one chromosome. So this is what it looks like. So prophase 1 of meiosis, here is your tetrad, okay? So they're lined up, loci to loci. Now, the whole thing, the whole process is called synapsis. So you got to keep these terms straight. And I know we name everything, but it's a pair of homologs. Okay. Now, look, I like it when they do electron microscopy. Now, the centromeres, look at the little wastes. Okay. So you can keep those straight. They form the little X's, the chiasmata. And then at the chiasmata, that's where the swap occurs. So you literally just swap that chunk. It could be a tiny little bit. It could be a bigger chunk. It's random, okay? It's different every time, every single time. And so they form the chiasmata, and then by the time you get to anaphase, look, you can see the little bits, anaphase one, you can see the little bits have already been swapped. And remember, homologs separate but not sisters in meiosis one. And in anaphase two, your sisters have separated. Some of them can be completely intact. Again, remember that crossing over is random. So you might have no crossing over that happens. That's as equally likely as, 1 to 4 events. And then here is the daughter cells. So you can have some recombinant and you can have some that are not recombinant. So there's possibilities here. Now, random fertilization is the last mechanism for genetic variation. Random fertilization just means you got an egg and it's got all those possibilities, right? It's making me raise my hand. I don't want it to raise my hand. Maybe it'll lower my hand. If not, I'm going to leave it up. I bet I can lower my hand. Oh, but just gonna leave my hand up. Okay, so random fertilization. Sorry, my hand is up. Random fertilization, okay, just means that, when males make sperm, they don't make like one sperm. They make like huge amounts of sperm, like millions of sperm. So each and every one of those sperm has like all these possible genetic combinations. Like remember, it's that 8.4 million possibilities due to independent assortment, that 2 to the power of 23, that thing. Okay, then technically, guys, crossing over adds even more possibilities than that, even more than that. All right, so that's crazier. But then you've got egg with the 8.4 million possibilities of genetic combinations. Then you've got the sperm, and you've got millions of those dudes. And then any single one of those that gets released can randomly fertilize the egg. And so, how you figure out what combination will actually come together, it's like rolling the dice in Vegas. Okay. So, the way you do this probability, and that's all it is a probability, we'll do more probability next week when we start doing Mendelian genetics, is you just take 8.4 million times 8.4 million, again, plug that into the old calculator, equals about 70 trillion diploid possible combinations per kid, per parental, same two parents, per parental unit. So if you and your sister come out as identical twins and you were born in separate days, separate pregnancies, that's a 1 in 70 trillion possibility. Do you see what I'm saying? Like, you probably look a little bit like your siblings, but you're not identical twins because the odds of that happening are 1 in 70 trillion. Okay. So each zygote has a unique, air quoting, unique, because I guess it is possible, 1 in 70 trillion genetic identity. And again, this is the source of genetic variation within our populations. This is the source of natural selection. This is the source of diversity in our world. It's the thing that binds us, and it's the thing that makes us so crazy different. And so it's the thing that selects out traits, and it's the thing that weeds out bad traits for the environment, and it's the thing that selects good traits for the environment. I often think about what made polar bears evolve from regular terrestrial grizzly bears. And it has to be their environment, right? Like as terrestrial bears moved into polar ice capped areas, they had to have stuck out like sore thumbs because they had dark fur. They couldn't feed very well. They couldn't sneak up on prey very well. And so mama polar bear, mama grizzly bear had one day, lighter color baby bears. And those baby bears didn't stick out as like sore thumbs. And they were able to sneak up and hunt a little bit better. They were able to obtain prey a little bit better. And then when they had babies, there were probably some really dark ones, there may be even lighter ones. And then over evolutionary time, out popped a really, really light one. And those were the most successful ones. And then over evolutionary time, they became the most successful. All of these weird traits that, match their environment or have some trait that seems so bizarre to us fit for some unique purpose and worked. And the traits that didn't work got weeded out. And so that's natural selection. It results in the accumulation of genetic variations fit for that environment. And in another environment, they may not work, and oftentimes they do not. All right, that is the end of this lecture. I'm going to say lower hand because that's really going to bother me. Okay, that is all for this lecture. I'm going to hit the end of this record button. End recording now.